Hello friends, good evening. This is Dr. Subrat Kumar Mohanty. Today we shall discuss about DNA ligase. DNA ligase is an enzyme that facilitates ligation. Ligation is the joining of DNA strands together through the formation of phosphodiester bond. The discovery of DNA ligase in 1967 by Richardson, Gellert, Lehman and Horwitz laboratories was a watershed event in molecular biology. DNA ligases are considered as the sine qua non of genome integrity. DNA ligase facilitate the joining of DNA strands together by catalyzing the formation of phosphodiester bond between the hydroxyl group which is present at the 3' end of one DNA strand and the 5' phosphate group of another strand. Many DNA ligases repair single strand breaks in double-stranded DNA in living organisms. There are few forms of DNA ligase which also repair double-strand breaks. Next point is ligases work with both sticky ends and blunt ends. You know that restriction endonucleases produces mainly two types of ends. One is sticky end and other is blunt ends. DNA ligase facilitate the joining of both blunt ends and cohesive ends. But in case of ligation of blunt ends, the concentration of the DNA ligase should be higher and the reaction conditions should be different. For the process of ligation, the enzyme DNA ligase requires an energy source. The energy source is ATP in bacteriophage and in eukaryotes, whereas in prokaryotes, NAD+ is the source of energy. Now we shall discuss about the mechanism of ligation. In this example, this is one strand of DNA, this is another DNA strand. This DNA strand provides 3' hydroxyl group. This DNA strand provides 5' phosphate group. Now the function of the DNA ligase is to catalyze the synthesis of a phosphodiester bond between this 3' hydroxyl group and this 5' phosphate group. So how this reaction occur? You know that for ligation, an energy source is needed. In bacteriophage and eukaryotes, ATP is the source of energy, whereas in prokaryotes, NAD+ is the energy source. So consider that ATP is the source of energy. So first of all, ATP will undergo hydrolysis to form AMP and inorganic pyrophosphate. Inorganic pyrophosphate will undergo hydrolysis in the presence of enzyme pyrophosphatase to form two inorganic phosphate. This reaction releases energy that is used for the process of ligation. At the same time, AMP will form a complex with the enzyme ligase. So here there is the formation of AMP ligase complex. The covalent bond link AMP with the epsilon amino group that is present in the lysine amino acid residue. This lysine amino acid residue is present in the active site of the enzyme ligase. So AMP ligase complex will be formed. After this AMP group will be transferred to this 5' phosphate group. This AMP group will be transferred to the 5' phosphate group. At last, nucleophilic attack will occur and a phosphodiester bond will be formed between the 3' hydroxyl group and 5' hydroxyl group. This is the mechanism of ligation that is catalyzed by the enzyme DNA ligase. DNA ligase is very much useful in genetic engineering for ligation of DNA strands. It is used for the construction of recombinant DNA molecules. When we are inserting a foreign DNA into a vector, this foreign DNA must be ligated with the vector. The E. coli DNA ligase and T4 DNA ligase are used in genetic engineering. But at present, the DNA ligase that is obtained from T4 bacteriophage is more widely used. DNA ligase play an important role in DNA repair and recombination. T4 DNA ligase is an efficient enzyme that repairs single-stranded nicks on double-stranded DNA molecule or double-stranded RNA molecule or even on RNA DNA hybrid. In genetic engineering, DNA ligase is useful for construction of recombinant DNA, for example, for the construction of recombinant plasmid. Friends, this is about DNA ligase. Please share, like and subscribe this video. My YouTube channel is Sam Online Class. Thank you very much. Bye bye.